Wonderful. Well, good morning, good afternoon. I'm Ben Lanai, based in Palo Alto, and I've been an AMN patient for about a decade. And I love to chair this session because it is really about what's going on in industry, the amount of interest we're getting from biotech companies to develop therapies for ALD and AMN. And when we started ALD Connect in 2013, we literally had only one company working on our disease. And now we have a cluster of companies who are well-funded, have great teams who are working on our disease. So a lot of hopeful and optimistic news on that front. So today we'll hear about several of them. First, I have to give the usual disclaimer, the legal disclaimer that we are not giving medical advice in this session. If you seek medical advice, you should see a physician, and we do not claim to prevent, diagnose, or treat a disease. But this is informational only, and I hope that it will be interesting. So before we start getting into each of the company's programs, I wanted to give a little bit of a refresher on drug development and specifically clinical trials. And I hope that it will be interesting to look at some of the concepts and the terminology for clinical studies, human studies. I myself was very involved in the Minorex trial. I consulted on the protocol design. I participated in one of the FDA meetings, and I believe I was the first patient enrolled at the Stanford University site. And I gave some feedback to the team on how to help improve the patient experience. I'm also currently consulting for another biotech company working on another neurometabolic condition and helping them with their design for that particular disease. So I think, you know, to start, drug companies basically discover drugs. You know, they discover what eventually will become candidates. And so they can do it, you know, with standard chemistry. More and more companies are using computational approaches, are doing drug discovery based on artificial intelligence. But the reality is that at some point you have identified a target and you declare a candidate. And that means that you basically put a stake in the ground and you say, you know, I have this agent and I think it could be used against this disease. And so at that point, after this target has been identified and curated and the candidate is declared, you know, you continue doing a bunch of tests, basically assays, and eventually we'll do animal studies to look at effectiveness in a live organism. And of course, those can be mice, zebrafish, and all the way to primates, which are very much like humans. And at some point, of course, you say, okay, now I need to try this in humans. And so I need to do a clinical study with humans to understand if the drug actually is safe and it works. So, and in parallel, companies deal with regulators. So in the US, the Food and Drug Administration, in Europe, the EMA. And in the US, what they do is they file an IND or investigational new drug application where they explain what their drug is, what it aims to do, and how it will be applied. And so the gold standard for human studies is still a study that is random, placebo-controlled, and double-blind. So random means that You know, basically patients are assigned either the drug, the active compound, or the placebo randomly. It's blind to the patient and to the study team so that nobody is biased. The placebo is a way, of course, to see you know, if people who are not taking the drug, how they're evolving compared to those who are taking the drug. And of course, observe very carefully any difference in patient outcomes. So a drug company will find sites. They will partner with clinical centers to actually conduct the study. So each site will enroll patients, will have a study team, and will follow the protocol in terms of administering the drug or the placebo, and then making sure that it's safe and observing the results. One big thing in the protocol design, of course, is exclusion and inclusion criteria. So who is admitted into the trial and who cannot participate. So for example, with Minorex, primary endpoint was around gait, walking speed. And so people who were completely wheelchair bound, for example, were excluded. You had to have still the ability to walk either by yourself or with an assistive device. But since it was the primary endpoint, you could not be completely immobile. You could not participate in the study. So exclusion and inclusion criteria, and often also are excluded people who have other you know, conditions, comorbidities, people are taking other drugs, you know, anything that could kind of blur the results. Then for the study, you have to define your objectives. So what are you trying to do? And specifically, what are your endpoints? So typically a primary endpoint, secondary endpoint, maybe some patient-reported outcomes. So again, for Minorex, it was primary endpoint was walking speed. Very important secondary endpoint was postural stability, balance, sway, you know, observing those things. And then patient-reported outcomes around, you know, bladder function, energy, fatigue, appetite, sleep, and mood. You know, and it could be just about anything, but you have to clearly define what you're looking for so that when you get the results, you see if your drug can be claimed to have been effective. Obviously, you're looking at safety, and you have to define adverse events, or things that could go wrong, and what is a threshold, you know, of gravity. So that you say, you know, we have some issues here, we have some toxicity that's unacceptable. You have to look at target engagement. So target engagement is a complicated word to say, you know, is the drug actually through its mechanism of action, is it, you know, triggering a chemical reaction or a cellular reaction, or is it getting delivered to the right tissues, the right organ, and then, you know, causing a measurable change in the chemistry of the body, and of course match to the disease, so we can claim effectiveness. And then companies look at what's called pharmacodynamics and pharmacokinetics, which is really how the drug circulates in the body. So you could summarize it saying, what does the drug do to the body, and what does the body do to the drug? And you look, you know, at how the drug is basically processed by the body. Then finally, in a trial, you look, of course, at duration. For AMN, it is generally considered to be a sort of neuromuscular, neuroskeletal disease. And so the FDA insists on studies that are long, unfortunately. So, you know, almost 2 years to really look at the evolution of the patient against natural history and say, you know, over this pretty long period of time, we have a noticeable difference that actually proves something. You have to look at dosage. So what kind of dose is safe, but also promising to be effective. And there are different designs with that. So there are trials where you have a single ascending dose or a multiple ascending dose. And you really play with dosage, and you can even adjust dosage based on tests from an individual patient and how they're reacting to the drug. And then to conclude, you know, why does a trial end prematurely? Sometimes it does. So one thing is called futility, and literally it's futile to continue the trial because the drug obviously doesn't do anything. And so you terminate the trial, you know, and that's happened, for example, in Alzheimer's. You know, there's been many failures where trials have been discontinued before their expiration. Another one is adverse events, severe adverse events. So, you know, there is some kind of toxicity. And, you know, we saw that a little bit with some of the kind of the second tier coronavirus COVID vaccine candidates. You know, things were put on hold or even terminated. And then occasionally, and that's a wonderful scenario, is overperformance. So a drug is so effective, almost in a miraculous sort of way, that, you know, you stop and say, okay, it obviously is having dramatic improvement in patients. And so we're going to the next stage and accelerating things. So these are all the considerations for a trial. And I hope it kind of sets the stage for what you're going to hear about today. We have some exciting candidates. Many of them are going to human studies in the next 18 to 24 months. And these are some of the considerations that are on all these individuals' minds as they architect their trials and try to refine their protocols to bring those drugs to the bedside. Thank you so much for your attention. And at this point, I will turn it over to Uwe. Again, Uwe, you're muted.
Good evening from Europe. I work for Minorix Therapeutics. We are based near Barcelona, but I'm based in Switzerland. So good evening from a nice summer evening here in Switzerland. I wanted to give you a brief overview on leriglitazone, on our study. And I am sharing my screen. I hope you can see it. Ben, can you confirm you can see it?
Yeah, we can see it.
Okay,
Uwe.
very good. I just put up the slide view. Okay, so here we go. Now our compound is called leriglitazone, or it has had a previous name. It's a laboratory code MIN102. Now, what is leriglitazone? It is a so-called PPAR gamma agonist. Now, what is that? PPAR gamma receptors are a family of receptors or binding sites that are inside the cell, at the nucleus of the cell, and they are involved in many biological processes. So one is it enhances the effects of insulin, which you know is a hormone that is deficient in diabetes, but it has far more effects. So it protects cells against insults. It promotes regeneration of cells that are dying or are already in need for rescue, and they have also effects against inflammation, which is very important. As you know, AMN can change into the other form with inflammatory brain lesions. Now, actually, leriglitazone is a metabolite. That means it is one of the molecules that is generated when the mother compound is metabolized. And the mother compound is called pioglitazone. It has the trade name of Actos. It is a very well-established medication to treat diabetes. And that means we could also refer to quite a few safety data from pioglitazone. And actually in diabetic patients, this leriglitazone, which is the metabolite called M4, contributes to a lot of efficacy of pioglitazone. We chose this over pioglitazone because it penetrates into the brain better, and it has a favorable safety profile because the metabolism is less complex. Now you may ask, why don't I just get a prescription of Actos, of the diabetes medication? We have experiments that tell us that if you take pioglitazone, you have to take 5 to 10-fold the dose that is actually approved for the treatment of diabetes. So that is, while it is in principle possible, it is practically not possible to reach that high exposure to pioglitazone. Now, this was our study that we completed, and we announced results, and also we have published that now at conferences. So thank you, Ben, for laying the groundwork. It was a so-called double-blind placebo-controlled study. That means patients received either placebo or they received active treatment. But in order to make it more acceptable for patients, with the perspective to be on placebo, to be on an ineffective medication for 2 years, patients had a likelihood of 2 to 1 to be on active treatment, while in many conventional studies, it is a 1-to-1 randomization. So you have only a 50% chance to be on active treatment. Patients who completed the 2 years are and have gone into a so-called open label extension. That means each and every patient is then receiving active treatment with leriglitazone. And we had studies in Europe and the United States. So Ben is right, he was the first patient at the Stanford site. Overall, 116 patients randomized. And if there are patients here on the call, we really wanted to thank you for your cooperation, for your patience, and for your compliance with the study procedures, which was not easy during the pandemic. And the open label extension is actually ongoing currently for patients. So we had a couple of visits, initially more frequently with safety examinations, pharmacokinetics, that looked at how many or how much of the compound went into your blood. We looked at the plasma levels. There were options to adjust the dose to the optimum for each patient. And then in half-year intervals, we had also study visits to read out on efficacy. Now, this is a bird's eye view on the results. As Ben already said, and I hope you can see my cursor, health authorities usually nail you down to define a primary parameter by which you read out the efficacy. And that was not easy for us because 4 or 5 years ago when we planned the study, very little was known about what happens in natural history in this disease. And we could resort to some so-called natural history data, that is observations at patients, just how they progress with the disease. And we used the 6-minute walk test that measures how long distance you can walk in 6 minutes. And here in the table, you see what came out of the study. So either there was no difference, or leriglitazone was even worse than placebo, but you can see that in no case, on no parameter, this happened. And then health authorities usually request you to define a so-called significance level. That is here the so-called 5% level, where you have to show whether or not actually your drug works on this parameter, the distance in 6 minutes walk. Now, overall in the population, it looked a little bit better, but it didn't look significantly better what health authorities require. However, we have now some analysis later on that showed in various models that actually the effect was dependent on age and time since symptom onset. And we could identify a subpopulation. These are patients who are of younger age and have less years since onset of symptoms that are really benefiting on this parameter. But what also came out is on the so-called key secondary parameter. This is body sway. This measures balance, your ability to maintain balance and posture. That in many of these conditions, actually there were very clear effects. Now the abbreviations here refer to EC as eyes closed, which is a position that is quite challenging for patients to stand and maintain balance. And FT and FA means feet together or feet apart. That means you have to stand on a plate that measures the excursions of your body with your feet either together or feet apart. And feet together is a very challenging position if you have some problems with balance. So here in this cluster of measures, there was clearly a better effect on drug. And also here we had two very complex rating instruments where investigators look at the entirety of your neurological symptoms. They're called SS-PROM and EDSS. This is a composite measure of various neurological symptoms, the so-called STEP score. And this is the ambulation score that measures, again, your ability to ambulate. What also very much appealed to us, and I'll show you details later on, is an event that no one hopes to happen, but unfortunately it happens in a proportion of patients, that cerebral lesions are seen, and then they may progress. They may even progress to inflammatory form, which is a very bad sign. And here, this is the so-called LERS scores. This is a score that radiologists give to lesions in your brain. So they score lesions in your brain comprehensively. And on these two outcomes, we clearly saw effects in the population. We also saw some interesting trends here in the so-called global evaluation. This is one, how clinicians rate your overall improvement, and here how patients rate their own improvement. And this is where physicians rate the severity of the disease. Interestingly, this is an analysis that we performed later. There were a couple of patients in the study who were not able to perform the 6-minute walk test either without walking aids, or they had to change to a higher degree of walking aid during the study. So from maybe 1 cane to 2 canes, or even from 2 canes to a walker and so on. And here we saw that actually significantly more patients in the control arm, in the placebo, required this change in walking aids. So overall, the picture looks very favorable and very interesting to us. And this is the next slide here. It shows you what I told you about cerebral lesion progression. So the sites were looking at MRIs of the patients in yearly intervals after 1 year and after 2 years of treatment. And they looked whether there was progression of cerebral lesions or whether actually lesions occurred. And also, they looked at the overall clinical picture of patients and said, yes, there is unfortunately progression in the MRI, yes or no. And you see that none of the patients, of the 77 patients on leriglitazone treatment, showed this progression of lesions, whereas there were 6 patients on the placebo treatment who showed this progression. And 4 of these 6 patients showed actually incidence of inflammatory lesions after starting treatment. And 2 of these 6 patients showed either incidence of a new lesion or growth of pre-existing lesions that were not inflammatory. And that is a very highly statistical significant outcome. That means it is very unlikely that this could be induced by chance. And here, this is the other one that I said is the comprehensive score that radiologists assign to cerebral lesions when they look at the MRIs. And also here, you see that more patients on placebo showed progression on these lesions than on leriglitazone. And again, that is statistically significant. So again, it is very unlikely to cause by a chance effect. And here, this shows you the change from start of treatment. The red lines are those patients who received placebo, and the green lines are patients who received active treatment. And you can also see from eyeballing in these patients that there was clearly less progression of these lesions in the MRI in the active arm. Now, regarding safety, it's a typical effect of all these so-called PPAR gamma agonists that initially they're called edema, cause edema, that is swelling of the limbs. Some patients also said they had a swollen face or around the eyelids. Weight gain is an effect of this whole class of compounds, and also increased lacrimation, so you have more tear flow. But what I show you here is that it doesn't skyrocket, this weight gain. So here the blue line is how the patients on leriglitazone developed over the 2 years of treatment. And this is % change from the start of treatment. And the gray line is the patients on placebo. So clearly see there's more weight gain caused by leriglitazone than on placebo. But we did have quite some weight gain also in the placebo patients. But when patients switched to the open label treatment, so these patients had already received 2 years of leriglitazone treatment, and you can see that weight doesn't only stabilize, but it goes down. No surprise, patients here were changed from placebo to leriglitazone, so they showed an increase in weight gain, but again, you see that weight gain is not only stabilizing, but is going down. So there were some patients who left the study for weight gain, but I looked at the effect of that, and I can say that it is not just caused by patients leaving the study. So it's not so an enrichment of those patients who didn't have weight gain. So whoever is on the study, the good news here is it can be managed. It doesn't go up infinitely, but it stabilizes or even goes down, and sometimes you need a dose reduction, but that is fair enough. Now, I also wanted to give you an outlook on the other study that we are conducting. It has the acronym NEXUS. This is a study in boys 2 to 12 years old. This is a study that is so-called open label. That means all the boys receive leriglitazone treatment. There is no placebo. And the boys are observed in a very similar fashion. As I said, all are on active treatment, so initially more safety visits. At 24 weeks, we have a short-term efficacy readout, and at 96 weeks, the definitive readout. And the efficacy readout is based on MRI of lesion progression in the MRI, or of progression of clinical symptoms. And this study is open to recruitment, as I said, in boys 2 to 12 years old who show initial cerebral lesions in the MRI. Now, this study is only ongoing in Europe, but for international patients, we provide travel assistance and accommodation as needed. Here are the 4 sites. 2 are in Germany, 1 is in Spain, and the 4th one is in Paris. So, as I said, in principle, all boys are welcome to join that study. And we did have international patients already on that study. Yeah, this is all that I wanted to show today.
Okay.
And happy to take questions.
Wonderful. We're going to get to questions later because I want to give everybody a chance to present. So next, we're going to have Rohan Gandhi, who runs clinical affairs for Audubon Therapeutics in San Diego.
Great. Thank you, everyone. Ben, can you hear me okay?
Yes.
Okay, perfect. Thank you. I do have some slides, but before that, I just thought I'd spend a couple of minutes just introducing myself to those that I haven't met. So good morning, good afternoon, and good evening to those in Europe. I am Rohan Gandhi, and I lead scientific and medical affairs at a company in San Diego in California called Audubon Therapeutics. And on behalf of all my colleagues at Audubon, it is a true privilege and a pleasure to speak with all of you today. So firstly, a big thank you to the organizers of the ALD Connect Bootcamp for inviting us today to talk to you about Audubon Therapeutics and about ABX002, which is our small molecule oral CNS preferring thyromimetic program, which I'll tell you more about as I present the slides. And these are for patients suffering from adrenomyeloneuropathy or AMN. I've been working in the biotech space for the past 2 decades. I've been with Autobahn since the formation of the company about 1.5 years ago. This was at the end of 2019, early 2020, just before the pandemic started, at least in the US. I had joined them from another rare disease company called Kiniksa Pharmaceuticals, which was based in the Boston area. So I've been working in the rare disease space for a few years now. And truth be told, I wish we could be meeting in person to share our stories and to introduce ourselves live. But I hope that the content and discussion, today will give you some perspective and show you the progress we've made since we've launched Autobahn Therapeutics as a CNS-focused company. And I'm really looking forward to the ALD Connect annual meeting later this year, which I hope will allow us an opportunity to meet in person. I want to share a verbal update on our clinical development path for ABX002. As you well know, there's currently a real lack of approved treatments. And as part of this community, I sincerely hope that the regulators view the data that Minorix has presented and Uwe, very nicely kind of just presented to you in a favorable manner. So there's an option for the patients out there. But currently there's really a high unmet need, which allows all the companies such as Autobahn Therapeutics and my other colleagues, the ability to really advance our programs fairly rapidly. And what we're planning is a single ascending dose study, which will be followed by a multiple ascending dose trial. And this will initially take place in healthy subjects. This will be followed by a phase 1b portion of our trial, where we will study the effect of our compound ABX002 in adult male patients with adrenomyeloneuropathy. And I'm happy to share with you that we've had initial conversations with the FDA, and they have agreed to the broad concept that we proposed to them. And as our trial progresses, we'll continue to have a dialogue with the FDA to agree on acceptable endpoints for the single pivotal trial. Our focus will be on 2 aspects. That's biomarkers, as well as the functional motoric endpoints. Okay, I'd like to share my screen now, and I hope you're able to see my slides.
Yes.
Okay, perfect, thank you. So we are a company that is innovating the next generation treatments of CNS disorders. And so we have a brain targeting chemistry platform, which is based on this prodrug technology that enables quite a broad pipeline of novel therapies. And the targets that we're going after on the top right-hand side are based on human genetics and have a very strong biomarker-enabled clinical development strategy behind them. The whole concept that we're going after is to drive regeneration in the central nervous system, in the brain. So all our lead programs, including ABX002, harness endogenous mechanisms to restore that function that has been lost. And as I mentioned a few moments ago, we're actually advancing to the clinic fairly quickly. Our lead program, ABX002, will be beginning human studies later this year in a couple of months. And we've had some conversations for an efficient path to a new drug application in 2025. Our plan is to leverage our technology to generate 1 new IND per year. And I think Ben described what an IND is. It's basically an investigational new drug application to the FDA. As you can see here from the slide, this year we're solely focused on adrenomyeloneuropathy and for moving ABX002 into the clinic. We've already generated a very robust in vitro and in vivo data package, and we'll be starting our first in human trials in the next couple of months. Behind this program is a separate program for patients that suffer from multiple sclerosis. And we're calling this program ABX003. This program is a first-in-class remyelination therapy, and we'll be beginning IND-enabling studies next month. And then behind these 2 programs, we have additional programs that will be driven from our discovery platform. A simple cartoon to try and explain how our prodrug technology allows these molecules to actually get into the CNS. So this cartoon depicts the brain and the blood-brain barrier, and by evolution, we've evolved to have a blood-brain barrier that limits the ability of these charged small molecules from entering the central nervous system. And this is, like I mentioned, an evolutionary conserved process that allows prevention, that prevents access to these foreign molecules that are typically negatively charged or positively charged. Our technology takes some of these molecules and converts them into a drug that doesn't have any charge, so it's neutrally charged. And this simple chemistry actually allows these molecules to passively diffuse across the central nervous system and be available in the brain. And these compounds have been designed so that these prodrugs are actually seen in the brain by an enzyme called fatty acid amide hydrolase, or FAAH. And when this enzyme sees the prodrug, it cleaves the prodrug, thereby releasing the active drug in the CNS. We're actually quite excited about the potential for ABX002 to provide potential benefit for patients suffering from AMN. And I won't go through the slide on the left-hand side because this is basically talking about what you already know, which is what are the genetic underpinnings of this disease, basically ABCD1 being dysfunctional, leading to a toxic buildup of these very long-chain fatty acids outside the peroxisome. And so what we're doing is we're actually having a dual mechanism. One is the ability of our target to directly promote genes that are responsible for remyelination of both central and peripheral neurons. And the second is actually to drive expression, to increase expression of another transporter protein called ABCD2, which functionally can complement the defective ABCD1. So in this cartoon, what I'm showing you is ABX002 will actually come into the cell, will bind to its receptor in the nucleus and drive the transcription of several different genes that are responsive to thyroid hormone. One of those genes is ABCD2. And even in the situation where ABCD1 is dysfunctional, we're able to increase ABCD2, and we hope that this will allow these increased very long chain fatty acids then to get into the peroxisome and get beta oxidized and be removed. Okay, that's all I wanted to say about the company. I just wanted to show you one or two more slides about this interest that we have in really understanding the journey that you as a patient goes through. And so we've been doing some work with some colleagues to really understand the patient journey. I just wanted to share that with you real quick, so you're aware that this is actually going on. So working in the rare disease space actually brings about a lot of challenges. And one of them is really to understand what is the journey of the rare disease patient. And so what we're trying to do is we're trying to understand the patient journey better via 3 consecutive projects, with the ultimate goal of really shortening the time to diagnosis, improving the patient and caregiver experience throughout the patient journey, and ultimately really using these insights to help design patient-reported outcomes for clinical trials and to make these clinical trials better. So these 3 projects are a patient or disease landscape, which we completed at the end of last year. What's ongoing right now is a patient journey. And then we're also working with Swan Bio. So Asif, who will be talking to you later, we're working with them to develop a novel patient-reported outcome as well. I just want to tell you a little bit about the patient journey work that's ongoing and what the objective of this is. So really the objective of the patient journey is to provide a really good understanding for us of the patient and the caregiver experience. You know, those that are living with AMN, what is the natural history of the disease? What is your journey through the healthcare system? And you know, what's your emotional journey, the moments of truth, the pain points, the moments of joy or satisfaction. And what we're hoping to get as an outcome from this are a couple of key points that are listed here. Number one, to understand the natural history better. What is the burden of the illness? What is of the cost burden, you know, prior to diagnosis, and then once diagnosis actually happen, because we know that there's a lot of misdiagnosis that occurs in this case. It's very important for us to understand what your physical and emotional journey are through the healthcare system. And then finally, you know, who are the key influencers and the decision points that might positively or negatively impact the clinical trial of ABX002 in this case. So, we've got this effort to really understand, what your needs are. And once we understand that, we can better design our clinical trials to make sure that we're providing what you are requesting. So thank you for making the time to listen to us. I appreciate that.
Okay, wonderful, Rohan. I'm sorry we're getting a little bit squeezed on time. So I will go next to David and ask that we keep it succinct in the interest of time. I'm very sorry.
I will try. Thank you so much. Can you hear me?
Yes.
Okay. So I'm David Moeller. I'm the Chief Scientific Officer at a company called Poxel. We are a small biotech company based in France. I'm actually based in Boston. Can you see my screen okay? Ben, can you see the screen okay? You're on mute.
Yes, we can see the screen.
Okay, so I'm going to quickly describe to you two independent programs that we are actively pursuing for AMN, both of which could potentially be in the clinic within the coming few months. These programs actually came out of some of the work we've done in other disease areas, including diabetes and liver disease, and they're focused on two different technologies, one of which is pretty closely related to what Uwe described for Minorix. We call these deuterium-stabilized thiazolidinediones. They're also related to pioglitazone, as Uwe described. And the second class of molecules are AMP kinase activators, which is a very different system that seems to be involved in regulating lipid metabolism, inflammation, and so forth. So I'll start with the first one, which is the deuterium-stabilized thiazolidinediones. As I mentioned, our molecules are closely related to pioglitazone, similar in some way to what Minorix is doing. But unlike pioglitazone, which works largely through PPAR gamma, our molecules work through other pathways that don't activate PPAR gamma and therefore have much more limited potential for things like weight gain, but still retain a lot of the anti-inflammatory and neuroprotective activity that we've seen with other molecules, including pioglitazone. And like pioglitazone, our molecules are also very closely related to leriglitazone, the compound that Uwe described, which showed promising results in patients with AMN. The other platform, as I mentioned, are molecules that activate an enzyme called AMP kinase. And this is a very interesting drug target for a variety of diseases and has benefits demonstrated, at least so far in animals, to improve lipid metabolism, to reduce inflammation, reduce tissue damage, and so forth. And we've actually shown for the first time in the last few months that this approach works in patients with liver disease to reduce the amount of fat in the liver, which could be related to what's going on in terms of fat accumulation in tissues with AMN. These mechanisms, these approaches fit very nicely with what we know about the molecular pathophysiology of AMN and ALD, because as you all know, the disease is associated with long-chain fatty acid accumulation in various tissues, spinal cord in particular, as well as inflammation, mitochondrial dysfunction, and cell death. And these mechanisms have the potential to address those fundamental aspects of the disease. So I'm just going to show you a couple slides to illustrate some of the exciting preclinical data that we've developed using both cells from patients as well as the classic animal model, which is the knockout mouse that lacks ABCD1, the gene that's also missing in humans. And with our pioglitazone related molecules that also have reduced PPAR gamma activity, we see very strong effects, both in patient cells to suppress very long chain fatty acids, to induce ABCD2, that's something that Rohan highlighted as an additional mechanism. And we also see in vivo in the animals that there's a very strong reduction in very long chain fatty acids in the brain and the spinal cord, as well as additional benefits that I won't have time to explain today. A similar profile on this slide is what we're seeing with the other group of molecules, the AMP kinase activators. And in this case, there's also an additional rationale that's in the literature. But what we've shown is a similar profile with regard to human cells and in the animal model, where we can reduce long chain fatty acids, where we can induce ABCD2 and also have additional benefits that I won't be able to discuss with you today. So what are we doing? We basically have two new classes of oral agents. These are oral once daily therapeutics. Both of our lead molecules have already been tested in clinical trials for other diseases. In fact, over 100 patients in each of the two cases. The lead molecule for AMP kinase has successfully completed multiple clinical trials in healthy subjects as well as in another disease indication. And the same is true now for the lead molecule in the deuterium stabilized TZDs, the 065 molecule. So we have the opportunity to take one or both of these molecules forward directly into larger studies in patients with AMN, and we're actively working on developing a clinical plan and a strategy to pursue something along those lines to begin within the next few months. So I'll take any questions later on, but I really appreciate everything that you all are doing to help support what we're doing in industry so we can work together to really solve this very difficult problem. Thank you.
Okay, thank you so much, David. Okay, and now Asif with Swan Bio.
Yeah, thank you, Ben. Can you hear me well? I'm outside today.
Yeah, we can hear you and we can see the slides.
Fantastic. Let's go to the next slide. Good afternoon, everyone. This has been an informative event for me, and I want to thank Ben, Kelly, and ALD Connect team for the organization, and thank you for inviting us. It's a true pleasure to speak with you all. I am Asif Packer. I work at Swan Bio. Swan Bio is a small biotech company based in Philadelphia. We are developing AAV9 gene therapy for AMN. I want to give you a short introduction to gene therapy. Can you go to the next slide, please? Yeah. Gene therapy introduces a working version of the faulty ABCD1 gene into the cells in charge of creating ABCD1 protein. This is done by using a vector, which is often derived from a virus, but the viral genes have been removed. So only the therapeutic genes are delivered into the cells. The modified cells then produce the protein that was missing or defective prior to the treatment. This vector can either be delivered outside of the body, ex vivo treatment. That's the right side of this slide. This is the approach that's taken to treat cerebral disease by Bluebird Bio. Or it could be done by the vector that can be injected into the cells. That's the right side of the slide, left side of the slide. This is in vivo treatment. I'll be talking about this in vivo treatment, that is the treatment modality for AMN. The AAV9 vectors carrying the working copy of ABCD1 gene in this modality is injected directly into the CSF. This is the fluid that surrounds the spinal cord and brain. With this injection, AAV9 vector will deliver the functional copy of the gene to the cells of spinal cord, and this will provide cells the opportunity to produce healthy ABCD1 protein to slow or stop down the AMN disease. Next slide. This is not a new modality of treatment. AAV9 gene therapy is an approved therapy for patients with spinal muscular atrophy. Similar to AMN, SMA or spinal muscular atrophy is a disease of neurons. In these patients, motor neurons are affected, and AAV9 gene therapy can slow down or stop the disease. Next slide. A team in Boston tested the AAV9 gene therapy in the mouse model of ALD. After the treatment, the healthy ABCD1 gene was observed in the cells across the whole spinal cord of the mice. And this led to the reduction of very long chain fatty acids. This work prompted us to develop the gene therapy for AMN patients. Next slide. I hope I'm not hurrying up. I'm going faster to make sure we finish on time. So our program we're working on this treatment modality in AMN has two types of studies. First one is the treatment study, where we will investigate the AAV9 gene therapy. And currently we are completing animal studies that are needed to start the first in-human study. I will reach out to you once we are ready to enroll patients in this study. The second study is the natural history study, which helps us understand the progression of the AMN disease. This will also help physicians and regulators to decide about the efficacy and safety of the gene therapy. This study has been started. This has been started earlier this year. In Europe, the study is ongoing in Amsterdam. And if you're interested, you should contact the Amsterdam Medical Center. Germany will soon start the enrollment too. In the US, this study will be conducted in Stanford, Utah, Boston, Baltimore, and Florida. These clinics will be ready to enroll in the fall of this year. In addition, as Rohan briefly mentioned, we are in collaboration with Autobahn to develop a patient-reported outcome measure with the University of Rochester. This will be an AMN specific endpoint that could be used in the future clinical trials of AMN. It will not only be for gene therapy study, but for all treatment studies. So keep an eye out for calls from University of Rochester. They will reach out to you through ALD Connect or ULF. I thank you all, and thank you, Ben, for the opportunity. And I'll take the questions now.
Okay, wonderful. Thank you, Asif. I know you had to rush through your wonderful content. So really appreciate you being a good sport about that. Don't know if we have time for a couple of questions, Kelly, or if we need to wrap up. But there were a lot of questions about minorics, a lot of interest. Can we still enroll in the open label extension? So that was definitely one question. When will it be approved? When will it be commercialized in the US? And can, in terms of clinical effect, can it reduce cerebral lesions once they've manifested? I assume, early stage cerebral lesions, or is it a purely preventative effect? So 3 different questions for you, Uwe.
No problem. Yeah, I can address it very briefly in one thing. So first of all, question was, what are we doing now? Now we are preparing the documentation to approach regulatory agencies, that is in Europe and in the United States. It is a huge amount of work. It's literally hundreds of pages where we have to summarize reports, where we have to summarize our view, what we think. But we are very close to approaching regulators. So it will be this month and next month that we are talking to the European and the US regulatory agencies to obtain their view, what they think. Do they share our view that we believe there is benefit for the patients? Although, as I said, formally, we have missed what we had predefined as the primary outcome, the walk test. But we think that in this rare disease, without available treatments, regulators may also look into other effects than the 6-minute walk test. For the time being, there's no other study, so there's nothing open for patients to join except for the study in these boys. The other question was, now what can leriglitazone do on brain lesions? Now, I'm very cautious to say that. I think there are very encouraging signs, but as you saw, it is 6 patients out of 116 where there was progression in the patients who were not treated. So this is a very small number, and you cannot make very firm conclusions on that. But also taking the current experience that we have from the boys study together, in my opinion, there are clearly encouraging signs also on ameliorating this inflammatory component, which is very scaring, of course, for patients. But as I said, I say that with a big word of caution, because there is also spontaneous stop of that progression, even in untreated patients. So now with these few patients that we have as experience, it is difficult to conclude anything firmly, but I'm encouraged, and we are continuing with the study in boys.
Okay, one final question with a quick answer. Maybe I will ask Rohan, potentially Asif, to answer. But question about, are these drug candidates all for early stage AMN, or can men with advanced AMN, late stage AMN benefit from those potential therapies?
Maybe I can jump in real quick. Ben, thanks for the question. Right now, the way we're approaching the clinical development is it's going to be available for all men at all different stages of the disease. But of course, as new data come, and as we learn more about what Uwe's team at Minorix kind of saw with the effect of their drug in different patient populations, we're certainly keeping our minds flexible in changing that. But right now, we're just thinking anyone over 18 should be available. Thank you.
Okay, wonderful.
And Ben, really, I can quickly say that we're planning to use gait and balance as our outcome measures. And for that reason, patient has to be able to walk and stand by themselves to complete these assessments. So there will be some limitations on severity of the disease that patient may have to enter these studies. But we will learn more once we have the protocol and design in front of us. Thank you.
Yeah, we have basically the same strategy at this point. A little premature to be too definitive about it.
Okay, wonderful. Well, we're going to wrap up, and I want to thank all the panelists, the audience for the participation, and of course, Kelly and Kathleen for organizing this wonderful session. So thank you so much and have a wonderful weekend. Take care.
Bye, Ben.
Thank you.